Hi, my name is Igor. I'm an undergraduate student from Universidade Federal do Rio Grande, and I'm gonna present my research entitled Genetic Diversity and Populational Structure of the Olive Ridley sea turtle, Lepidochelys olivacea, in southern Brazil. The olive ridley have a circumtropical distribution, nesting in tropical waters with migratory circuits in tropical and subtropical areas. And in Brazil, the nesting regions are between the south of Alagoas and north of Bahia, but the main rookeries are at Sergipe and north of Bahia. But we have sporadic nesting at Ceará e Rio Grande do Norte and Espírito Santo e Rio de Janeiro states. It's a species that uses both coastal and oceanic waters for feeding and development. And it's classified as threatened by the Red Book of Endangered Brazilian Fauna of the Ministry of the Environment and as vulnerable by the Red List of Threatened Species of the IUCN. The coastal waters of South Brazil are important foraging and development grounds for all the 5 species of sea turtles that we have here. And the olive ridley stranding have increased over the last decade at the region, and this increase was evident from 2010 onwards. Despite the high worldwide abundance of olive ridley, little is known about their genetic characteristics on the Atlantic Ocean, and there is no information on their genetic diversity in foraging areas. And these genetic studies can help uncover some important information, such as diversity and adaptive potential and structural connectivity among populations. So, in this context, our objectives were to evaluate the genetic diversity, population structure, and possible natural regions of olive ridley turtles from foraging area in southern Brazil. 59 olive ridley samples were collected between 2013 and 2019 through beach monitoring from the middle of the coast of Rio Grande do Sul to the limit of the Brazil with Uruguay. The mitochondrial DNA was extracted using self-extraction, and an 800 base pair of the mitochondrial DNA control region were amplified by PCR. Haplotypes were classified according to the NCBI gene bank. The haplotype and nucleotide diversities and structure analysis were calculated in Arlequin, and a haplotype network using the Median joining method was constructed in PopArt. For evaluation of populational structure, we used data available from olive ridleys from other locations in the Atlantic. Our preliminary results indicate that the olive ridley aggregation in southern Brazil has low nucleotide and haplotype diversities, as we can see on this table, and only 2 haplotypes with 1 base pair difference between them were found, both already reported for this ocean. And this low number of haplotypes is in accordance with other olive ridley populations in South Atlantic. This low genetic diversity indicates that the population may not be expanding and may have gone through a possible loss of genetic diversity due to a bottleneck, and this could also be associated to a recent founder event in the Atlantic Ocean. In relation to the structure analysis, low and moderate genetic differentiation was observed in the rookeries in the southwestern Atlantic, and a large structure was observed between the southwestern and the southeastern Atlantic rookeries. These results indicate that the olive ridley populations within the southwestern Atlantic are somewhat connected, but have some structure with the eastern Atlantic. This structure may be to the distance between areas, and the Atlantic Ocean could be acting as a soft biogeographical barrier. The predominance of haplotype F among sampled turtles indicates that the feeding area likely receives individuals mainly from Atlantic rookeries, since this haplotype has only been reported for this ocean basin. With these preliminary results, we can conclude that the southern Brazil aggregation has low genetic diversity. In an ocean basin context, the populations present high connectivity, especially in the southwestern Atlantic. Some structure is observed with the eastern Atlantic. And the feeding area probably receives individuals from rookeries in the northeastern Brazil, which have a high nesting number, are geographically close, and possibly linked via favorable ocean currents, such as the Brazil Current. I would like to thank these institutions and the organization of the Genética 2021.